16.1 goes over a system of differential gene expression, which will lead to different cell types in a multicellular organism. So when we think about embryonic development, you have fertilization. So egg and sperm come together, or the sperm fertilizes the egg, and at that point, the zygote is made of two cells. And then it will divide and divide and divide and ultimately become a very different multicellular organism. And what I mean by different is inside of that organism, the cells are very different. So a lung cell is different from a skin cell, different from a stomach cell, etc. So we learn this process of how gene expression is regulated to bring about those different types of cells that make up a multicellular organism. There are three main processes which lead to an organism having so many different types of cells. The first type is regular old cell division that we learned about with mitosis. This will increase the number of cells in an organism, but the catch here is that although we're increasing the number of cells, the cells are the same. So the purpose of that cell cycle was one cell making another cell which is identical to it. So this is a large number of identical cells being produced with cell division. The second type is known as cell differentiation, and this is a process where cells are becoming specialized in their structure and function and being organized into different kinds of tissues and different kinds of organs. The third type of process is morphogenesis, and this is where there's a physical process of giving the organism its actual final shape. One way that a cell knows how to differentiate and have its cells turn into different types of cells is cytoplasmic determinants. If you look at the diagram, you can see the large circle is an egg that's unfertilized, and then you can obviously see the sperm cell. Once the sperm fertilizes the egg cell, if you look at the distribution of the molecules that are inside, some of them on the left are brown circles, and then some on the right are green triangles, indicating different types of molecules. These could be proteins, these could be different organelles, mRNA. They can be anything that the cell needs, but they're clumped differently. And when you have the zygote, which again is a fertilized egg, and it has two nuclei inside, it's going to divide and then divide and divide. And the quantity of those molecules inside will ultimately determine or help to determine what type of cells the cell will actually become, and then mitosis will occur for identical cell division. The other way that cells know how to differentiate from one another is with induction. And this is a process where cells are triggered to become different or to become the same. When you have neighboring cells together in an embryo, so again, looking at the diagram, I think the diagrams can help a lot. You can see there's an early embryo of 32 cells, and you have contact between them. There's cell-to-cell signaling going on, where molecules from one cell are influencing the neighboring cell to most likely grow and differentiate just like it. So having target cells with signals being received, that is another way that cells can differentiate. Determination of a cell is the irreversible series of events that leads to cell differentiation. And to remind you, differentiation is the actual process of where cells will become specialized. If you look at the diagram, it's showing you in red different words that you should be familiar with. Totipotent, or it looks like totipotent, that is where the cell can make or divide into any kind of cell type, any and all that are present in the organism. The next set, when we are further along in development, whether it's a human fetus or a later on embryo, the cells are known as pluripotent, and they can become almost any type of cell in the body or differentiate to any type of cell. And then finally, when we actually have cells that make up our organs or organ systems, at that point they are known as unipotent. They are only capable of making or differentiating into the same exact cell as it already is. This slide is, or this diagram is a little bit confusing to understand, and it's on page 324 in your book. This picture is showing you that there's two things going on in order for differentiation to occur, and determination, which has to be the beginning step. So there's genes in DNA that will obviously be transcribed into mRNA, which ultimately down the road through translation will make certain types of proteins. So when you look at this diagram, the first thing that needs to occur is determination. So what type of cell are we looking at? If you look in the top middle, you'll see a master regulatory gene known as MyoD. That is a master gene. That has to be turned on for transcription and mRNA, a specific mRNA, to be made. At that point, it is determined what type of cell this cell is going to be. Myoblast is a type of muscle cell. So now the master gene is on, and we for sure know that this is going to be a muscle cell. Now then, the differentiation needs to occur in the specialization of the cell. So for differentiation, we're going to need a series of other genes in different kinds of mRNA to be made to further differentiate this muscle cell. So the first thing is the master gene is turned on, again, determining what we're working with. Then we have other genes that are specific to being a muscle. They have to be later turned on, and then we have the process of a specific type of muscle cell. As an embryo is developing and making more cells, a lot of the times these cells are produced in excess in that embryo. So some cells need to be programmed to cell death, known as apoptosis. And one of the proteins that help assist to chop up the DNA organelles and all the cell parts or cell components, those are known as caspase proteins. If you look at the diagram at the bottom, and this occurs in humans as well, what you're being shown is a mouse hand, mouse paw, and you can see it. The first picture on the left doesn't even look like a hand. It doesn't really have its digits. They're there, but you just don't see them. There's been too much tissue made around the actual digits. So the cells in between need to undergo apoptosis, so that way we can see the space in between the actual fingers or digits. Sometimes cells other than embryonic cells need to be programmed to die. An example being shown to you is a human white blood cell. Most likely it's been infected or damaged beyond repair, so it needs to be sent to death. One part that occurs, if you look on the left, that's the white blood cell, and on the right is the same or different white blood cell undergoing apoptosis. Now the one on the right has these vesicles branching off of them. The process which forms those vesicles is known as blebbing. And the blebs will be engulfed by different types of cells that come around and engulf them. And then eventually, thanks to these scavenger cells, there is no more damaged white blood cell left. There are molecular cues in an egg or an embryo that controls pattern formation. What pattern formation is setting up of the body plan, whether it's a head, tail, left, right, back, front, as a result of the cytoplasmic determinants that the mom has and inductive signals that the cell can receive from neighboring cells. So fruit flies are a perfect example in how a fruit fly develops from the egg in the mom to being laid and into a larva, which it'll eventually turn into an adult. So looking at this summary, the first thing that we have is the egg at the top right. The egg is surrounded by a follicle, which are support cells, in the female or the mom's ovaries. Then we have a mature, unfertilized egg, and at this point the egg has enlarged because it has received nutrients from the nurse cells that were surrounding it. And the nurse cells, I think shaded by like a peach color, they eventually shrink because they are being depleted. Part 3, we now have a fertilized egg, and then it's going to be laid by the mom outside of its body, obviously. And then part 4, the egg is going to develop into a segmented embryo. And part 5, the embryo will turn into a larva, and then there's a pupa stage, and then we eventually have an adult fruit fly. Now that we understand a bit about Drosophila, which is a fruit fly, and how they develop from egg to adult, it's important to know what genes are responsible for pattern formation. Again, the body plan or the organization that the fruit fly body is going to have. Homeotic genes are the genes that control pattern formation, control the spatial organization of the organism. Hox genes are an example of homeotic genes. These Hox genes code for Hox proteins, which specify the characteristics or position of correct structures in the correct places of the fruit fly's body. So if you look at the diagram, on the left is what's considered typical or a wild type, and you can see the head of the fruit fly, the eye, the antenna. When the Hox genes or homeotic genes had a mutation, then we had body plan displacements. So now the mutant, which has again a mutation taking place in the Hox or homeotic gene, you can see now instead of the antenna coming out of the middle of the head, we have legs instead. So anytime there were mutations in these homeotic genes, they caused a severe misplacement of body structures. As the embryo is developing, there can be an uneven distribution of substances that can help to establish an embryo's axes. These are known as morphogens. And if you look at the diagram, there's a gene known as a bicoid gene. So please remember the bicoid gene is going to be transcribed into bicoid mRNA, which will make certain kinds of bicoid proteins. So what this bicoid actually is, it's a maternal gene. And anytime there is a mutation in bicoid alleles, it can result in that mutation that we saw where a fruit fly won't have a proper anterior. So first, two words that you should be, actually 4 words you should be familiar with. Anterior is like the head part of the organism. Posterior is like the butt or end part. You should also know when we're looking at like a fruit fly, all along the top of it is going to be known as the dorsal top, and then like the belly underneath would be known as the ventral part. So please make sure you understand the difference between the 4 different types of body plans. But what we're talking about with this is the front face, the anterior part, where we saw on that, I think it was the last slide, where we saw the legs growing out of the face. So back to the bicoid alleles. When we have mutations in both of these, we see that the embryo develops two tails and an abdomen and no head whatsoever. So this suggests that the bicoid gene is essential for setting a normal anterior part of the fly. And it goes back to having a heavy concentration of this protein in that anterior part of the embryo. If we don't have that, then we're going to have mutations down the end. So again, substances building up and developing in the embryo in proper concentrations can contribute to hopefully having proper body plans. And if something is not distributed evenly, then we're not going to have what's typical in terms of the anterior, posterior, or dorsal or ventral part.